All right, thank you, everybody. We're moving on to the next session, which is we've got 5 phenomenal speakers lined up for this morning. They're going to tell you about the research that they've been working on. And I want to give a huge thank you to Anna Yashchenko, who is a graduate student in Anna Stepanova's lab. And she, as a graduate student, is a volunteer to moderate this session. So she'll be introducing the speakers and doing her best to keep them on time. And as a reminder to the speakers, Anna, where are you going to be sitting, do you think?
I'm going to be sitting at that table.
She's going to be sitting over here. We asked the speakers to give, full disclosure, we asked them to give an 8-minute presentation and 2 minutes for questions. So at 7 minutes, Anna's going to stand up. So everybody in this room is going to see that and realize where you are in your presentation. That means, speakers, that means you have 1 minute left to speak. At 8 minutes, she's going to start creeping towards the speaker to give them the heads up that they really need to wrap it up. And depending on how much time is left, Anna's going to make the judgment call. Do we have time for questions? If yes, great. If not, then what we'll ask is that everyone here, if you have questions for the speaker, please ask the speakers during the breaks. And with that, I'm handing it over to Anna.
Hi, everyone. I'm Anna. Thank you, Jeff, for introducing me. I'm a 5th-year PhD candidate. And I'm going to be introducing everybody's talks today. So first up, we have Natalia Duque-Wilckens in biological sciences, giving us a talk about mast cells.
Okay, hello everyone. My name is Natalia Duque-Wilckens. I'm in the Department of Biological Sciences. I'm a semi-new assistant professor, and I'm going to give you like a quick overview of what my lab is doing. So I've always been really interested in psychological health. And as such, you may think that I study the brain, right? And within the brain, I study neurons. But turns out now we know, like in the past 2 decades, that the brain actually interacts with the rest of the body, including, for example, the gut in a process that's well known as the gut-brain axis. But within the brain, there are also other cells that are not neurons. These include immune cells, such as microglia and mast cells. While microglia are very well-studied cells, mast cells are a little more mysterious, and that's why I'm kind of really interested in them. We also know that the brain actually controls or has the capacity to influence peripheral immune organs and immune cells, and that this, in turn, can affect how the brain works. So I'm really interested in studying how the whole system interacts to drive behavioral and physical health, and how this in turn is affected by the environment. So that's why I'm interested in mast cells. And as you can see, this is a very pretty cell, but I'm not only interested in mast cells because of that. Actually, mast cells are really fascinating cells. They express a variety of different receptors that allow them to respond to a huge variety of different stimuli, ranging from toxins and stress to neurotransmitters, bacteria and viruses, and parasites and allergens. In turn, mast cells can respond to this stimuli by releasing, again, a plethora of biologically active mediators. These include neurotransmitters, microRNAs, growth factors, cytokines, and so forth. So this, together with the fact that masses are distributed throughout tissues in the body, including the brain, makes mast cells ideally positioned to modulate multiple physiological processes. And indeed, although we didn't know much about them until recently, we mostly knew about them about their relationship with allergies. Now we know that they play a variety of roles in peripheral systems. And this also means that mast cells could play a fundamental role in brain-body communication, and this is why I'm really interested in them. So in my lab, we have 2 main projects, both of them associated with mast cells. The first one is trying to answer the questions, do mast cells play a role in behavior? And the second one is trying to answer the question, could mast cells be a key link between early life exposures and lifelong susceptibility to physical and mental diseases? So for the first project here, I wanted to show you really a quick video. So to answer the first question, we started by comparing the behavior of normal mice with mice lacking mast cells. So I'm not sure if I can, oh, there you go. So this is a test called the social interaction test. And here you can see a wild-type mouse, a mouse that has mast cells, and how actively trying to figure out who is in that little cage, which is like that cage contains another mouse. However, when you look at mice that do not have mast cells, you see that although they seem interested, they look at it, they do not really approach. And this is a behavior that we call social vigilance, which is like an anxiety-like behavior. So actually, we were not the first ones to show that. Other labs, for example, Ray Silver's lab, have shown that animals lacking mast cells show increased anxiety. And actually, two recent papers in Nature show that mast cells are key in driving this behavior of avoiding foods containing allergens. However, we do not really know how mast cells are doing this. So the first question that we wanted to answer in our lab is, where are mast cells affecting behavior? Is it from the periphery or is it from the brain? So for this, we took bone marrow from wild-type mice, grew mast cells in culture, and then injected them back either in the brain or in peripheral tissues in mice lacking mast cells. So then we compared the behavior of these guys. We compared the behavior of wild-type mice, meaning mice that have normal mast cells, with those mast cell knockout mice, and those mast cell knockout mice receiving mast cells in the brain or in the periphery. So this is a test called the elevated plasmate test. It's a test that measures anxiety-like behavior. And here in orange shows you the behavior of the wild-type mice, showing that they spend a significant amount of time in the open versus the closed arms of this test. But when you look at the white bar there, which is the mast cell knockout mice, you see that they show like reduced time spent in the open arms. And this is again an indication of increased anxiety-like behavior. However, when you inject mast cells either in the brain or in peripheral tissues, this behavior gets normalized. And then we found the same thing with this other test called the sucrose preference test. As you can see, it's the same trend. The animals lacking mast cells show reduced sucrose preference behavior, but this is reverted somehow with injecting either mast cells peripherally or within the brain. So this is telling us that mast cells can affect how the brain works and how the behavior works, acting both from peripheral tissues or from central tissues. But we don't know how mast cells are doing this. So now what we're really focusing on in the lab is trying to figure out how this works. So one of the approaches that we're using is we're trying to do tissue-specific mast cell molecular profiling. So for that, we are using a model called TRAP, basically, which allows us to tag translated RNAs within mast cells in specific tissues. We're also using another technique or approach in which we would use transgenic mouse lines to be able to tag proteins specifically in mast cells, to then do proteomics and compare how mast cells in the periphery differ from those mast cells in the brain. Additionally, we're trying to figure out, by using the thyrotoxin-mediated specific cell ablation, what happens if we get rid of mast cells in the brain or in peripheral tissues? How does that affect behavior, and how does that affect brain cells and brain function? On the flip side, we're also doing gain of function. So we're kind of trying to do the same experiment that we did initially, except that now we're going to follow up with like a cellular and molecular analysis of the brain to see how putting mast cells back in the brain or in the periphery affects brain function and behavior. And lastly, we're really excited. One of my students is generating the first human brain organoid containing mast cells. Human brain organoids is a model of human brain function that grows in culture. Is a 3D model that structures itself. And because of the nature of these organoids, which are derived from ectoderm cells, they naturally lack immune cells. So they do not have mast cells, presenting a unique opportunity for our study. And I see you're standing, so I'm going to go really quick over this. The other question that we're trying to answer is, are mast cells possibly a link between early life exposures and lifelong susceptibility? And for that, and supported by CHGA at NC State, we're studying how early life exposure to flame retardants affect mast cell function and behavior later in life. So briefly, we found that exposure to flame retardants during the perinatal period decreases mast cell responses to allergen and infection-like stimuli. And we also found that this is not only in vivo, but also if we take bone marrow from these guys and grow mast cells in vitro, we see the same response, suggesting that whatever effects there are on mast cells during early life, they are acting at the progenitor level. So with that, I just wanted to thank all my mentors. Without them, I wouldn't have being able to be here. Also, Heather Pattisol, who was the first who decided to give me a gift of let me try to start studying these things. Also, the National Brain and Behavior Foundation for funding my studies of the mast cell knockout mice and wild-type mice. The CHG and also the CMI for funding a new project that we have in collaboration with Kurt and Jeff Yoder, trying to make a zebrafish mast cell reporter model. And obviously, all my lab members. There are some missing here, but I'm extremely thankful. And that's it. Any questions? Oh, and these are actually mast cells in the brain. As they are like the GFP positive mast cells. Can you see that? No. Is it? Oh, great. So those are the red are astrocytes and the green are mast cells, and you can see that they are kind of connected to each other. Yes.
Looked as if all of the treated mice were white versus the melanin hair. Is there an effect of pigmentation?
Yeah, that's a really good question. Actually, the reason why these mice lack mast cells is because they have a mutation in the c-KIT receptor, which also affects melanin production during adulthood. So yes, this model is not a very clean model, and that's why I was really interested in assessing whether these differences in behavior were due to lacking mast cells or something else. And that's why we put mast cells back, which turned out to be a really cool experiment. Yes.
Love your talk, by the way. I was curious when you were looking at one of your previous experiments, the re-injecting the mast cells. The mice that didn't have mast cells, I noticed is kind of interesting. It almost seemed like their population was kind of separate, like bimodal in the data. Did you guys kind of look into to see if there was a difference between the ones that had like the higher population versus the lower population on that set?
That's a really good question. We were trying to figure this out. We tried assessing whether we tried to do correlations with, for example, peripheral serotonin circulation or histamine. We didn't find anything. We're trying to figure that out. But yeah, it's definitely a bimodal distribution, at least in that behavior. And it's very clear.
We got one more question.
Yes.
So the mast cells are tied, obviously, to the immune function, which communicate with the MHC. The MHC is tied to odor production and odor detection. So have you looked at chemical signaling in your mast cell deficient mice?
Chemical signaling, like in.
It affects their ability to detect odors and to produce odors. So when you're seeing behavioral differences, it could well be mediated by chemical signaling.
That's a really good question. We haven't looked into that, but we do know that, for example, these mice have increased pain sensitivity. So I wouldn't be surprised that many other senses and perceptions are affected, not only their social motivation. Yes. Thank you.
Thank you, Natalia. Our next speaker is Kasie Raymann from Plant and Microbial Biology Department, and she'll be talking to us about honeybees.
Hello, everyone. So I'm relatively new here at NC State. I started actually about exactly a year ago. And before that, I was an assistant professor at UNCG. So I'm really happy to have stayed in North Carolina and to be here. It's been a really great year. So I'm going to talk about how we use the honeybees as a model system to study both beneficial and pathogenic microbes. So just a broad introduction. Bacteria play a major role in animal health, and I'm sure you all know that. It's been something that's been established for a while now. But bacteria and specifically evolve and adapt really, really quickly. And this gives them the opportunity to be able to adapt to different environments. And in fact, most bacteria are able to live in multiple environments, which is really interesting that they're so versatile. Because they evolve and adapt really quickly, different strains within the same bacterial species can have vastly different functional capabilities. So you can have some bacteria that are in the same species that can differ by up to 30% of their gene content. Microbial communities, of course, are providing really important functions for their host, but one big question we really don't understand is how do some of these microbes have the ability to persist and live within inside of a host, and some, on the other hand, do not. Some microbes can also become pathogenic under certain circumstances, and I'm going to talk about those today as opportunistic pathogens. And what I'm really interested in is what's driving the evolution of opportunistic pathogens, which are pathogens that don't rely on a host for transmission. So it really raises the question as to why they would evolve virulence and maintain it. So the overall question that my lab tries to address is just broadly what factors are driving the ecology and evolution of host-associated bacteria. This is a complicated question because, as you can see, microbes that live inside of a host have to deal with a myriad of different factors. So they not only have to deal with the host itself, so its immune response and its genetics and other factors, but they also have to deal with a lot of interactions with other microbes, and toxins and nutrients that come into the system. So the methodology we use in the lab, and I'm just going to talk about two of the major projects that we work on in the lab right now. One is when we're working with microbial communities. This is where our Honeybee model system comes in. So we're doing controlled sampling, that meaning that we can put honeybees back in the hive with a controlled age, and then we can sample them under different conditions and evaluate their microbiome composition. We also do inoculations in the lab, and I'll talk a little bit more about how we do that, where we can inoculate bees with different microbes and then study the function and the dynamics of those communities. And then we also do some serial passaging, where we're basically experimentally evolving microbial communities within a honeybee host. And we do all these things to try to answer questions about species level diversity within microbial communities, strain level diversity, as I told you, which can be really important, and then the genetic and functional diversity within communities. The second big project in my lab has to do with looking at opportunistic pathogens and how they evolve. And for this, we do controlled environmental experiments in the lab, so putting them with nutrients that we know or in conditions that we know, like lower oxygen, temperature, etc. We also evolve microbes in different conditions with different predators and in multi-species communities. And then we're also evolving opportunistic pathogens within our host model system, the honeybee. And our goal with this is to understand how all of these factors impact the genotypes of opportunistic pathogens, their phenotypic characteristics, as well as their virulence within hosts. So why honeybees? So first of all, they have a highly stable and really well conserved microbiome that's socially acquired from their nestmates in the hive. Their gut microbial community is relatively stable, so when we want to study the microbiome systems, usually in mammals, they're so complex that it's difficult to study them. But honeybees only have 4 core bacterial genera present in their gut microbiome. They can be reared germ-free and then inoculated with known microbes, and here I'm just showing you how we do this in the lab. So if we pull the pupae from their capped cells, before they're able to move their mouth parts, we're able to then rear them in the incubator in the lab, and they emerge relatively germ-free as you could get, an organism. We can also perform highly controlled experiments at really large scale. As you can see here, we can have thousands of honeybees in the lab at a time in these cup cages where we maintain them. And we can also, like I kind of alluded to earlier, we can bring bees in the lab, have them emerge and mark them, and then return them back to the hive and then retrieve them afterwards. And down here, these are just little bees when we hand feed them in tubes. We put them in these tiny little tubes and we get to hand feed them with pipettes. They're really adorable. So we also study honeybees, of course, because they're agriculturally important, and they're very charismatic. I mean, if you work with them for a while, you fall in love. So just to give a few brief overview of results from these 2 different projects that I told you we have going on in the lab. So first on microbial communities. So I know I just told you that honeybees have a really relatively simple microbiome community. However, there have been a lot of studies that have shown that there's extensive strain level diversity within the species that are in the honeybee microbiome. And that strain level diversity has been shown to correspond to a lot of different functional capabilities in honeybees. And what's interesting is that honeybees within the same colony, even though they might have the exact same species composition, if you look at the strain level, 2 honeybees can have a lot of different strains present in their microbiome. So we're really interested in understanding what's dictating this fine scale strain level diversity within individual honeybees. And so to do that, so far we have looked into several different factors, one being geography, another being age, host genetics, diet, and microbial interactions. So for geography, we sampled bees from different locations around the United States, and we looked at their microbiome composition, specifically strain level diversity, and we found that there was no, geography was not dictating the strain level diversity. We then looked at age, so we did this controlled study where we had bees of known age, and then we sampled them from the hive and looked at their diversity and found that that also wasn't dictating strain level diversity. So none of these factors so far were telling us what would give us the answer. So we thought, since honeybees also have multiple genetics in the hive, maybe it could be host genetics, but it wasn't that either. And then we have looked at some studies for diet, and we have a little bit of evidence that diet is playing a role in changing communities. And then we've also have some evidence that microbial interactions are also playing a big role. So for opportunistic pathogen stuff, we're basically going off of the idea of the coincidental evolution hypothesis that suggests that pressures in the outside environment can lead to virulence in pathogens. Specifically, one hypothesis that it's predation. So we evolved an opportunistic pathogen in the presence or absence of a predator. We're still evolving it, actually, but this is just the results after 60 days. So what we found is that in the presence of a predator, we had increased biofilm formation, grazing resistance, and virulence, and all those things were decreased without the predator's presence. We also found a lot of parallel evolution across our different lines, and surprisingly, those evolution events were present in genes that are in the same protein-protein interaction network, suggesting that they are tightly linked and that all of these microbes in different environments are taking kind of the same pathway to get to where they are. So this suggests that virulence and virulence-associated traits could be due to selective pressures by protists. So next questions, basically, we just want to keep studying what's driving community composition, understand how stable communities are over time, and look at if they have the same functions and what interactions occur. And then for opportunistic pathogens, we're evolving other opportunistic pathogens, not just the one I mentioned today, and trying to really understand how opportunistic pathogens evolve and adapt to different lifestyles. So with that, I would like to thank all my current lab members, some former lab members that were involved in the work that I discussed today, and then my current collaborators at NC State, but I'm new here and I'm really excited to start collaborations with others. And then, of course, all of my funding. Thank you.
We have time for one question. I want to ask two. Sorry, break the rules. First one, do you get stung a lot? And the second one is, I think bees definitely have different jobs within a hive. Have you looked into how having different roles in the hive might impact their microbial community?
Yeah, I mean, kind of indirectly, because age corresponds with their lifestyle in the hive. It's not a perfect proxy because they'll change depending on what they need in the colony. And it's a little more complicated. I can talk to you about it later. But so the age kind of correlates. So we couldn't really separate age from being a nurse or a forager, right? And don't get stung very often. I've been stung once this year. And when we're in the lab, the bees have no desire to sting you because they're not protecting their hive. So in the lab, they don't try to do anything to you. Just when you're in the colony and they're trying to protect their queen and their sisters. And I'll talk to y'all later if you want. Thank you.
Thank you, Kasie. Our next speaker is Claire Gordy in Biological Sciences, presenting on behalf of Biological Sciences Belonging Research Group on elevating marginalized student experiences in life sciences.
Thank you. My name is Claire Gordy. I use she, her pronouns, and I'm a faculty member in biological sciences. So I first want to thank the students who participated in the interviews that I'm going to be talking about today for sharing their experiences with us, along with acknowledging the funding from the HHMI Inclusive Excellence Program to Dr. Jane Lubisher that helped us carry out this project. So the work that I'm going to be talking about today was carried out by an interdisciplinary research team who are shown here. And so it's important to first recognize the ways in which our shared and individual positionalities inform the work that we are doing. So our team consisted of undergraduate students in the life sciences, as well as doctoral students in the College of Education, and faculty and staff from life and social sciences. Across the team, we are diverse in regards to race, ethnicity, immigrant status, sexual orientation, age, disability status, and economic status. And we all have a shared research interest in understanding how we can transform post-secondary STEM education to make it more anti-racist, diverse, equitable, and inclusive. So as I share this work today, I am not speaking on behalf of North Carolina State University or the UNC system, but I am sharing the results of research that is intended to elevate the experiences of our students. So I'm going to be talking about belonging, and it's important to begin by defining that term. Belonging is defined as a subjective assessment and feeling of connection and legitimacy as a valued group member in a social setting. And it is actually a fundamental human need. So if you look at Maslow's hierarchy of needs, belonging only comes after physiological needs. So that's food and water, and safety, that's shelter. And I think in general, we accept that students need to have their basic needs like food and shelter met before they are able to successfully learn. The same is true for belonging. So from the literature, we know that students who perceive that they belong have better academic performance, they have a stronger self-concept, and they're more likely to remain in their major and their institution. We also know from the literature that students with marginalized identities have lower sense of belonging in community. And the same is true here at NC State. So from some unpublished results from a survey-based study, we found that students from a number of different marginalized communities felt decreased sense of belonging within the Department of Biological Sciences. And interestingly, this was also true of graduate students in general as compared to undergraduates. We specifically saw that students with these marginalized identities feel less comfortable with both faculty and peers within the Department of Biological Sciences and in their STEM courses outside of biological sciences. And we found that 30% of our respondents had seriously considered switching out of their biological sciences programs. This is more common among first-generation students than other students, and some of the common reasons included lack of sense of belonging and lack of support. So how do we create an educational community where students feel they belong? We took a qualitative case study approach to this question. We used semi-structured interviews. We spoke with 27 undergraduate life sciences majors, and those interview questions focused on their experiences within their classes and within the Department of Biological Sciences, their feelings of belonging with their classes in the department, and their perceptions of the department's efforts toward diversity, equity, and inclusion. We then performed an analysis of the transcripts from these interviews using open coding guided by the multi-contextual model for diverse learning environments, or MMDLE, as a framework. And what that allows us to do is to ensure that we are identifying codes and then themes that really take into account all of the different contexts in which our students exist. So those include the communities of which they're a part outside of the university and their external commitments, along with their institutional context and its climate for diversity, which is made up of all of the interactions and experiences they have with their peers, faculty, and staff inside and outside of the classroom, along with the larger policy and socio-historical context in which the institution exists. So through that analysis, we identified three themes in the students' quotes, and those themes can be thought of as sort of recommendations that the students are making to us. Those recommendations include implementing inclusive pedagogy, improving co-curricular resources and support, and humanizing life sciences education. And it's important to recognize that these recommendations are not just for things that we individually should do in our classrooms or our research labs. They also relate to larger structural issues and policies that we can push for change. Throughout all three of these themes, we also found that students repeatedly emphasized that we need to approach all of these changes with authenticity and intentionality. So I only have time to share a few of their quotes with you today. And I am going to actually read through what they said and talk about it just a little bit. Under the implementing inclusive pedagogies theme, one student said that just mentioning inclusion statements on the first day of class, they're not like following through, especially with biology. If you're showing the anatomy of a human, if every single person that you're showing always has white skin, when I know that there's ways you can find a black and brown person, having that statement in your syllabus, but then not really committing to it down to like the small things are the ways that I've seen it. So this student is expressing to us that the inclusion statement in the syllabi in some of their courses is not coming across as being authentic because it is not aligned with the course learning materials that the instructor has chosen. So this is an opportunity for us to be more intentional in ensuring that we are choosing more representative materials for our courses. Another student mentioned, I'd also like to see more people of color and more people who look like me in the department, not just students, the people who work there, people who hold higher leadership, who are tenured advisors. I would want to see more diversity within that. So we know from the literature that having faculty and staff who are representative of the identities of the students is important to student success. What this tells us is that this is not an unconscious or implicit effect. This is something that our students are very aware of. Another student said that they had a group project last semester. It was over a kind of a long period of time. They felt like it made them find a place for themselves. And they're speaking about this being a group project and that helping them to build community during a pandemic. This is another opportunity for us to be intentional in designing class activities that are built to help build that community. Students also discussed things outside of the classroom. We had students who discussed our department getting their own counselor and how this made them feel valued as a student and that we are supporting their success. And then finally, a lot of students spoke about humanizing life sciences education, the importance of professors being vulnerable, being humans, sharing their own struggles with the students to make them comfortable for coming forward when they need help as well. So I don't have time to share any more quotes with you, but we have a lot of take-home messages from this that are actionable items that we will be able to share with you when this paper is published in terms of things that you can directly do in your individual practices in the classroom or in the research lab, as well as larger structural changes that we can push for. Going forward, we're interested in expanding the study to understand more about graduate student belonging. The students we interviewed were undergraduates, but from our earlier survey-based study, we know that the concerns of graduate and undergraduate students are different. We also want to understand how sharing these findings with faculty impacts their teaching and mentoring practices, and to understand how faculty perceptions of disability accommodations impact the experience, not just of disabled students, but of all students. All right, and I'm happy to take any questions. So we initially, so yes, I need to repeat the question. The question was, how did we identify the undergraduate students that we interviewed? So we started with a survey-based study that went out to the entire Department of Biological Sciences, and we had hundreds of respondents to that. And we then reached back out to students who had responded to that survey to see who would be interested in participating in interviews. And the students who were interviewed were all compensated for their time. Mm-hmm.
For thinking about this longitudinally, given the policy changes happening both federally and within the UNC system at large?
Yes. Yes. So, is there space or thoughts about continuing this work longitudinally, looking at changes related to the policy changes coming from the UNC system? And yes, absolutely. Yeah. Great. Thank you.
Thank you, Claire. And now we move on to our next speaker, Trevor Walker, assistant professor and co-director of the Tree Improvement Program, and he's going to talk to us about genetics in pine.
Hey, thanks a lot. I'm really excited to be here and present on some of my research, but I really want to take this opportunity to talk about the program that I work for because I'm really proud of it. It's the Tree Improvement Program at NC State University. This is our website, treeimprovement.org. Our mission statement is to make forests more productive through the use of genetics research and breeding concepts. And this is right so we never run out of wood. So we don't cut down the trees to make houses, make paper, and not have enough for future generations. And also to make the plantations that we have more productive so we can release pressure from those ecological, those cooler natural forests for other ecological reasons and personal enjoyment. So our program has been around for a while. The founder, Dr. Bruce Sobel, he founded other programs, tree breeding programs here at NC State and around the world. And he was really hardcore, right? He would actually climb the trees to access the female and the male strobili to conduct the breeding. He was a contemporary of Norman Borlaug and felt very passionately that we need a genetic resource in our forestry industry. And so our funding comes from about 40 different organizations. These are timber companies, these are state agencies, federal agency, USDA. And we use that support we get from them to leverage grant funding. And it's a cooperative. And the way that the cooperative works is that our members are responsible for planting experiments, measuring those experiments, and then getting the data back to us. And the Tree Improvement Program at NC State, we act as the researchers that design the experiments. We have 2 faculty. I'm co-director. Dr. Fikr Isik is the other co-director. We have 2 emeritus faculty. We have 3 full-time staff members. I used to be on the staff. And we try to keep at least 5 graduate students. And we're presently looking for graduate students. So the species we work with is the most important timber species in the southern United States and probably the world. It's loblolly pine. I'm sure you've heard of it. This is just a picture of a plantation, trees that are being grown for timber production. And we're talking about 1 million acres that are planted every year using genetics that came from the NC State program. We have the legacy of the program has a big fingerprint. This is 1 million acres every year that are being reforested with the genetic improvement that originated here. And the way that this gets out to the plantations is through seed orchards. They harvest cones from these trees that have been genetically selected. And right now we're in our 5th cycle of breeding, and we want to get those cones out as soon as possible. So this is an example of wild, non-improved loblolly pine on the left, so no artificial selection, the square one. And on the right is an example of a first-generation family. The only difference between the two is that we know that the mother of the trees on the right has good genes. That's what we identified through testing. Same deal, but here we have 2nd cycle versus 3rd cycle. And the way that we do this is through a recurrent selection program. It's not rocket science. It's just taking trees that look good, mating them with one another, and then taking their seed and putting them out in replicated, experimentally designed tests. And it's not rocket science, it's quantitative genetics. And so we're using the linear mix model to do the best job we can of taking the phenotype of a tree plus an experimental design and its pedigree or its genomic relationships based on DNA markers to separate the part of the tree that looks good because of its environment and the part that's due to its genetics. And so by spinning that cycle around faster using genomic selection or just doing a good job of our breeding and testing is really how we improve the livelihoods of the citizens of this country. The cooperative effort, I think, is shown best with this map that shows the trials that are established. We plan about 10 trials every year across the southeastern United States. This is a great data set for graduate students to work on master. This is one of the data sets I use for my PhD, many other PhDs, and there's more work that needs to be done. So phenotyping these trees and then choosing the trees that look good and getting them back into the breeding cycle. Another research area that we have is we can't just take the best looking trees. We have to, right? We need something else in a breeding population. It can't just be all about gain. We also have to balance diversity. And one of the way that we do that is we have a large population size that we breed at our breeding facility and orchard. And we use algorithms, mathematical algorithms to balance gain and diversity. And I'm proud of our program because we're one of the first to jump on this use. It's technology that we borrowed from the livestock literature, but also developed on our own. And it's another active area of research. It's how you balance gain and diversity. So we like to get our students at the breeding orchard to get hands-on learning. And looking at some quantitative genetic stuff, what kind of gain have we made? So on this graph, if you look at the 0 dotted line, that's where wild non-improved, no artificial selection started. And this histogram shows the gain in growth. It's a score that we call productivity, indicates how fast the trees are growing. It's a percent gain measure. So we've got about 30%, 35% gain in the second cycle. Then we choose parents to breed for the third cycle, choose parents to breed for the fourth cycle. And here we are in the fifth cycle. We've chosen parents to breed. And we've made tremendous gain. And we're looking forward to continuing that. Now, so that's typically using just quantitative genetics, phenotypes, and pedigree. Another active area of research is this fusiform rust disease. It's a native co-evolved disease, co-evolved pathosystem, and there is variation in resistance among trees. There has been demonstrated that variation, that resistance can be controlled by a small number of genes. And this is where we have had excellent success bringing in genomic technologies and DNA sequencing to do a better job of understanding the mechanisms of resistance. Right? Whereas for yield, it's a complex trait. So we depend on our quantitative genetics models. So right now, our ongoing research, it's detailed in our most recent annual report. I've got the link there, treeimprovement.org. We've got genomic selection projects. We have an exciting validation project at genomic selection where we've gone through a round of recombination to the next generation to validate the way that those markers are keeping their trait associations. We've got marker-assisted selection projects, mostly around disease resistance, plant pathogen interaction projects. We also study reproductive biology so that seed orchards can be more productive. And then if you think about that map of all those progeny tests, right, we could probably do a better job of including environnomic data, weather data, soil data, to partition the genetic effect out from the environmental effect of those trees. So that's all I have. Yes, thank you. Thank you for your attention. If there are any questions, I think we have time.
It's time for questions. Thank you. That's a great talk. The question is soil. When you assess the growth of these trees, are you also taking into account soil changes in response to accelerated growth?
So the question was, while we're assessing the trees, are we also assessing the soil to understand how that influences? And so, did I get the question right? So how not only are the trees growing faster, but how does that change affect the environment that they're growing in? Is there some sort of like circular feedback? And the answer is we have no idea. And this is what's called the random effect in your linear mixed model. And certainly there is, and I know there's exciting research to separate those things, but it's not something that we've looked into. Yeah.
but like, are you also looking at quality of the wood? You mentioned how long it takes for a tree to grow up. Out of curiosity.
Yeah, so the question was, how do we select our, what I would call selection targets, the traits that we select for? How much emphasis? So we use an index breeding value that's like a linear combination, 60% volume, 20% straightness, 20% rust. And we have about 40 organizations that are members of our co-op, and they come up with those numbers. They tell us, this is, we're farmers. This is what we need to be most profitable and to meet our stakeholders' needs. And you'll notice I didn't say wood quality in that. We have this funny dynamic in the United States now where the folks that turn trees into lumber are not the landowners, they're not the farmers. So farmers sell it to the mill. And so there's not a lot of, most of the emphasis is on yield rather than quality, which is unfortunate because that trait is so heritable. We can make the wood so much stronger if we apply artificial selection.
One more question.
Yes.
Yeah, I just wanted to comment that it takes a long time to grow a tree. So does the community change its mind over time while you're doing a generation? And how does that impact?
So the question was pointing out it takes a long time to grow a tree. And does the community that we're working with, do we change our mind before we can get anything done? And that's what happened with our third cycle. But most of it was because the initial plan was too ambitious. Foresters are a patient group of people. And so they're used to it. But that's why we have to spin this cycle faster using new technology, so that not because we change our mind, but because we have changing climates. We also have changing markets. There's the forest biomaterials department in our college is constantly finding new things to make out of trees. And so we need to be able to change tack and make the trees better for those new products. Thank you.
All right, so now, as many of you know, the GGA organizes summer contests for students and postdocs, and one of those is the GGA Paper of the Year Award, where students and postdocs can apply with their paper to justify why their study is impactful, and the person who is chosen gives a 10-minute talk at the annual retreat. So this year's winner, who will be giving the talk, is Daniel Sulis from Jack Wang's lab. And this is for his paper, Multiplex CRISPR Editing of Wood for Sustainable Fiber Production. So let's give him a big round of applause and welcome Daniel. Congratulations.
All right, so I'd like to start thanking the organizers for this invitation. I'm really happy in receiving this award. And today I'm going to be talking about some of the work we are doing in the Forest Biotech Group in Professor Jack Wang's lab. So forestries are really important ecologically, especially at this time of climate change. But also forestries are the most abundant biomass on Earth and a very important natural resource for our society. So for example, from wood, we can produce energy, we can produce timber, or we can produce a wide range of renewable bio-based materials, including fabric, fibers, paper, biofuels, packaging, and alternative fibers. However, the conversion of the wood biomass into some of those bio-based products can be really challenging depending on the wood composition, or more in particularly, depending on the lignin content in the wood, which is the major bottleneck for the industrial processing of wood. So how we can genetically improve trees to efficiently produce those bio-based products? Well, this is really challenging, as we saw in the previous talk, that tree genetics is highly complex, and we don't understand tree genetics as we do for model plants and crops. And also, as we saw also in the previous talk, the breeding cycles of trees can take decades, which basically limits the generation of new improved tree variants with traits of interest. So we need to find alternative to speed up the genetic improvements of trees. So in our lab, we're using a different approach. We're combining machine learning predicting algorithms with CRISPR-Cas genome editing technologies to identify and edit key genes related to traits of interest. So, and then doing so, we can generate trees with desirable phenotypic changes. So in our case, we're interested in traits related to the composition of the wood. So we can, for example, alter the composition of the wood, such as reducing lignin content, so the major bottleneck for the industrial processing of wood, and also increasing the fiber content, so increasing cellulose and hemicellulose content. So by doing so, we can basically make those conversion of wood feedstocks into different bio-based products, including fibers, biofuels, a much more efficient process. And of course, besides wood properties, we're also interested in growth properties. So we want to generate trees that can grow really well, not only under control conditions, but also in the field. Well, so CRISPR-Cas, they work as molecular scissors, generating double-strand breaks at the DNA. And those double-strand breaks, they are repaired by the cell machinery, but often those repairs are imperfect, leading to nucleotide insertions and deletions at those target sites and resulting in gene inactivation. So taking advantage of this approach, we added 10 different wood-related genes individually or in combinations in poplar and generated 174 different genome-edited lines of poplar. So 174 different genotypes of poplar. So we took those 174 genotypes, we planted in our greenhouse, about 272 trees. We let them grow for 6 months, and then we evaluate their performance in terms of growth properties and also in terms of wood quality. So editing some of those wood-related genes impacting the phenotype of the wood, as we can see in the figures. So we can see major changes in the coloration, ranging from white in the wild type to orange and red in some of our lines, which was a really nice indication in how powerful, precise, and efficient CRISPR can be in generating fast or inducing fast genetic diversity in forestry. But those changes, they were not only visual, but they did impact in the phenotype, in the chemical composition of the wood, as you can see in the plot in the right, where we could recover lines with very low lignin content. So in a reduction almost up to 50%, and significant improvements in fiber content, so in cellulose and hemicelluloses, which basically is optimal for industrial processing of wood. So we want less lignin and more fibers. Well, but how those alterations in the chemical properties impacted in the further downstream applications? So basically, in terms of fibers and paper, we saw that utilizing those CRISPR-edited wood, we can see significant improvements compared to the wild-type controls, as represented here, the CRISPR lines in the blue dots and the wild-type in the red dots. And those improvements can reach up to 40% at the industrial scale, which could lead to substantial financial gains in the pulp and paper industry, up to $1 to $2 billion per pulp mills. But besides fibers and papers, CRISPR can open up a lot of new possibilities and markets, such as in the production of renewable biofuels and chemicals. So we saw that utilizing CRISPR, we can enhance the production of biofuels and chemicals by 2.5-fold compared to the wild-type wood, and also it can increase some of the mechanical properties of other bio-based products, including some of nanomaterials like cellulose nanofibrils. But more important than increasing efficiency of the wood conversion to bio-based products or adding new traits to those materials is the environmental impacts that CRISPR can generate in forestry. So we basically doing a life cycle assessment, we saw that basically if the CRISPR-edited wood is implemented at industrial scale, it could decrease the emission of carbon dioxide by 31%, while decreasing energy consumption, water consumption, and chemical usage. So basically, this is the approach, the approach we're using in the lab. So essentially, using predicting models and CRISPR, we can add specific genes related to traits of interest, and then making the conversion of those bio-based products a much more efficient process and also a greener process decreasing the emission of carbon dioxide, water usage, energy consumption, chemical usage, and so on. Of course, there are some challenges and limitations we have to address using this kind of approach, such as how those plants are going to behave in the field. We so far have planted them only in the greenhouse control conditions. So we are currently running a field trial, and we're going to evaluate several traits, not only the wood quality, but interaction with the environment and many others. So as was mentioned before, some of those results are part of this publication in Science, republished last year. So if any of you are interested in knowing about our work in more details, please feel free to access the paper. So with that, I'd like to acknowledge my two mentors, Dr. Jack Wang and Dr. Rudolf Berengu, and also my lab colleagues and other collaborators across different departments at NC State and also across different universities. Oh, thank you so much for your attention.
I'm sure you get this question a lot, but I'm curious about mechanical strength of edited wood. Is it comparable to wild type? What is the composition effect?
So it was asked if editing the wood would affect the mechanical properties. So for the lines we have analyzed so far, we saw only in two some alterations in wood density and models of elasticity, but for the majority of the lines, we don't see major or significant changes in the mechanical properties. But it will depend of the genotype. So some of the genes we may even increase, some of the genes we can decrease a little bit wood density. It's really variable depending of the targets that you are editing with CRISPR. So the question was if those improvements were in lines that had harbor only one gene added or multiple genes added. So basically, we need more than one gene added. So the lines with significant improvements, they had at least 3 genes being concurrently added. So I got lines from 3 to 6 genes added, and those are the ones with the more improvements. So basically, more complex those genetic strategies are, or those genotypes are, more benefits we can get out of those. Yeah.
I was curious about carbon balance and carbon budget for this plant. So I noticed on one of your slides you mentioned that they had reduced CO2 emissions. And
Yeah.
one obvious place would be that that's going to the lignin. But is that actually like does the increased biomass actually fully account for the decreased CO2 emissions?
So the decrease in emission of carbon dioxide is because you produce less lignin, and so lignin is like a byproduct, right? So with lignin, what is done is lignin is burned after the industrial process of wood, nothing else. So if we generate less lignin, we burn less wood and we emit less carbon dioxide to the atmosphere. So that's why we see a decreasing in carbon dioxide emission.
We're out of time, so thank you.
Thank you.